Hi everyone, this is the fifth lecture for your chapter Cell. In the last class, we discussed at great length the structure, function and composition of cell wall. Okay, focusing on the plant cell wall, but we also discussed a few things about the fungal cell wall, and also we had already discussed in prokaryotes the prokaryotic cell wall, which is made up of peptidoglycan, right? So today we will be starting with the next boundary. Okay, what comes after cell wall is cell membrane. Cell membrane is seen in all living organism. How many ever living organism are there? You cannot point anyone out saying they do not have the cell membrane. Okay, cell membrane are also called plasma membrane. They're also called plasma lemma. All of these three mean the same. Okay, they have the same meaning. They mean the same membrane that is around your cytoplasm. Fine. So today we start with cell membrane or plasma membrane. Now before we can start, like always, let us discuss some history. Okay, so there were scientists Gorter and Grendel in 1925. They extracted lipids from your RBCs. Okay, this is a scanning electron micrograph of RBC. I had told you there were two types of electron microscopes: scanning electron microscope and tunneling. Okay, so this basically this will show you the outer structure of the cells. Fine. So from the RBC membranes, because RBC do not have any other organelles, okay, I'm telling you this first. RBC does not have other organelles, so it was easier to get pure cell membrane from RBC. So they extracted lipids from red blood cells and showed that lipids could form bilayers as well as monolayers. Okay, let us see how the lipids were. So they were actually what you call phospholipids. Okay, they did not know what kind of lipids there are and all, but when they extracted meaning to separate, okay, from the cell membrane of RBC, they found such kind of phospholipid. Okay, now this phospholipid can get arranged in any manner, right? How are they arranged to form a cell membrane? How do you know? So they can be arranged like this in a single layer, or they can be arranged like this in a double layer. Fine. Or they can be arranged like this. Fine. So there were a lot of possibilities how the cell membrane might be arranged. Okay, how the what you can say phospholipids in a cell membrane are arranged. There was a lot of possibility. So they said they could be in bilayer. Bi means two, layers means two layers. Okay, so here is the first layer on the top and here is the second layer. Right, now why are they arranged in this manner only? This later on we will find out that these tails are hydrophobic and the heads are hydrophilic. So that is the reason why a bilayer will always form like this. The heads will be outside and the tails will be inside. They can also form a monolayer. Monolayer is a single layer. As you see here, they form micelle. Micelle meaning the outermost cover will be of the polar head, and towards the inside there will be all the hydrophobic or non-polar tails. So this here is the single layer. Fine. Or there could be a liposome-like structure where you have again a bilayer, but it is forming a small sphere. Okay, so that is a liposome. So these were the different arrangements in which the cell membrane could be existing. Right. These were found from the RBC membrane by Gorter and Grendel. Biochemical analysis revealed that the cell membranes are made up of lipid, protein, and carbohydrate. Now, apart from what the previous scientist had said, they apart from having lipids, they also had proteins and they had carbohydrates. Okay, so if you take this RBC cell membrane and if you zoom it, you can see this is the lipid bilayer. Okay, and on the lipid bilayer you see a lot of protein is present. All of this other structures that you see are proteins, and the branches you see, okay, tree like structures you see on the top are actually carbohydrates. Okay, so the cell membrane is not only made up of lipids, it is also made up of protein and carbohydrate. How did we find out this? We found it out by biochemical analysis, meaning you separate the cell membrane and then you purify it and analyze it to find out that there is lipid, protein and carbohydrate present. The ratio of protein and lipid is different for different cells. Okay, this protein, how much protein is present compared to how much lipid is present, varies from cell to cell. Human RBC membrane, okay, for human RBC membrane, there is approximately 52% protein. Okay, there is more protein compared to lipids, which is only 40%. Okay, even from this diagram, if you see, if you just remember, okay, not the names, but the photograph, if you recall, then you will be able to understand that there is a lot of protein. Can you see how much protein there is? And compared to that, the lipids are very less, right? So the RBC membrane, important to know, it has 52% protein and 40% lipids. So now that we knew what cell membrane or plasma membrane was made up of, different scientists started giving their own models. Okay, because till now we did not have any way of finding out or by microscopic methods we were not able to see. Okay, because cell membrane is so thin, it is 7 to 9 nanometer thick. 7 to 9 nanometer is very small, and electron microscope was not very well developed. So what people did, they knew what it was made up of. They knew it was made of lipid, protein. Okay, so they started giving out their own models. They started assuming that it must be made like this, or giving scientific explanation saying that their model is the best. Okay, we'll see that now. The first model is sandwich model by Davison and Danieli. Okay, sandwich model by Davison and Danieli. So they said plasma membrane is made up of 3 layers. It is made up of how many layers? 3 layers, like a sandwich. You have 2 bread and in between you have some stuffing, right? So here also there will be 3 layers. A layer of lipid is sandwiched between 2 layers of protein. He said there is one layer of lipid which is sandwiched between 2 layers of proteins. Okay, so there will be 2 layers of proteins surrounding one layer of lipid. Each protein layer is 20 angstrom thick. Okay, so these protein layers are angstrom thick. How much angstrom? 20 angstrom. Okay, another thing, they said this protein is in globular form. So you can see these spheres, globular or globe means circular, spherical, right? So these proteins are present like this in their globular forms. Okay, and they were 20 angstrom thick. After these protein layers comes the phospholipid layer, which is 35 angstrom thick. So total, if you do 20 plus 20 is 40 plus 35 is 75. Okay, so depending on the protein thickness, it could be 75 to 100 angstrom thick. That is what they said. Fine. Phospholipids are amphipathic molecules. Now let us understand what phospholipids are. This will be a quick revision of the biomolecules. Fine. Phospholipids are amphipathic molecules. Amphi, wherever you hear the word amphi, it means two. Okay, it is doing function of two. There you use amphi, like amphibians, right? Amphibians are animals which can live on land as well as in water. So here amphipathic molecules are the molecules which have a polar side and a non-polar side. Okay, or polar meaning hydrophilic, they are water loving. Okay, so they have a polar head or hydrophilic head and non-polar tails or hydrophobic. Phobia is fear. What are they fearful of? They fear the water. So hydrophobic tail. Clear? So this is the structure. The hydrophilic head is made up of phosphate, what you see here, PO4 minus. Then you have glycerol. So that glycerol, let me just draw real quick. A quick recap. Okay, glycerol is a 3 carbon molecules. It has 1, 2, and 3. It has 3 OH groups. Okay, it has 3 OH groups. Pardon my terrible drawing. I'm not able to draw well here. Okay, with help of a mouse. Fine. So it has 3 OH groups. With one OH group, there will be a phosphate group. Okay, PO4 minus or 2 minus. And with the other two, there will be fatty acid chains, which are nothing but hydrocarbons. Okay, so you have fatty acid chains, which are shown here. If you see very clearly, this is one fatty acid chain and this is second fatty acid chain. So these fatty acid chains, this portion is what forms your two hydrophobic tails. Okay, and phosphate as well as this structure forms the hydrophilic head. Okay, which is this. Fine. Okay. Hydrophilic head binds with protein layer. Now protein, which is mostly hydrophilic in nature, some of them are hydrophobic, which we shall discuss. So this hydrophilic head will be in touch with the protein. What you see here in dark colors are all hydrophilic heads, which are in touch with the proteins. Clear? How are they in contact with the protein? So they are either forming hydrogen bonds or they are forming ionic bonds. Ionic bonds are formed when there are positive or negative charges. Okay, and hydrogen bonds are formed when there is a delta positive and a delta negative charge. Clear? Let us take an oxygen atom. Oxygen can make hydrogen bonds. Okay, and there is a hydrogen which is partially positive. So there will be a sort of bond forming between these two. Okay, it is semi-covalent bond. It is not completely covalent bond, but it is somewhat similar. So this partially negative, partial positive, the bond between them is what is called hydrogen bond. It is the same bond that you see in between water. Okay. Hydrophobic tails. Now all of these hydrophobic tails are attracted to each other by van der Waals force. Van der Waals force is a weak force. Okay. And they are attracted to each other by van der Waals force. Clear? So this was the model given by Davison and Daniele. Okay, they said there was a protein layer, protein layer on each side. Okay, and in between there was a lipid sandwich. Fine, the lipids were present in the between. The proteins were unevenly spread according to them. They were in form of this globe. Okay, and yeah, so the phospholipids were present in lipids. The polar head was in contact with the protein and the non-polar tail were in contact with each other. Now just for your information, there are different types of phospholipids. Okay, this is the glycerol structure. 1, 2 and 3 carbons. 3 carbons have 3 OH groups. OH groups form different bonds. Okay, here R2 is showing fatty acid and it is forming ester bond, COO bond. Okay, COO bond is ester bond. So esterified. Okay, so there is here R1. So there will be first fatty acid, then here is the second fatty acid. Now let me just clear this mess up. This is the first fatty acid, second fatty acid, and on the third carbon, instead of fatty acid, a phosphate group is attached. And to this phosphate group, you can attach different kind of molecules, okay, different X groups. What are these X groups? They can be phosphatidylcholine. Phosphatidylcholine is basically like this, okay, it has O, it has CH2, CH2, then it has one N, and to this N there are three methyl groups, CH3, again here CH3 and here CH3. So this is basically the X group. Okay, it is shown here as OCH2CH2 N and CH3 whole thrice. So this is how they are actually arranged. Fine, erase, fine. So that is called phosphatidylcholine. This group is called choline, and when it gets attached to this group, this region, it is called phosphatidyl. This group is phosphatidyl. Okay, two fatty acids and one phosphate group with glycerol backbone is phosphatidyl, and the X group is choline. So phosphatidylcholine. It is commonly called lecithin. Now this is homework. Okay, whatever you bring from outside, whatever food packets you have, try and find out the ingredients. You will find the ingredients at the back. Okay? If you have chocolate, well and good, find on the back of chocolate, you will see something like this written. They write soy lecithin. Oh, this is N. Okay, here lecithin. So find out what soy lecithin wherever you find it. Okay, on what food items you can find soy lecithin. Fine. So common name is lecithin and scientific name is phosphatidylcholine. Then there is phosphatidyl ethanol amine. Ethanol amine is OCH2 CH2 NH3. Okay, so this is the ethanol amine. Ethanol because CH2 CH2 and this was actually OH. Array. Okay, CH2 CH2. Then you have here NH3. CH2 CH2 NH3, and on this side you have OH. Okay, so carbon chain length, if you see, there are two carbons. So this is definitely ethane. It has an OH group, so it is ethanol, but it also has an amine group, so it is ethanol amine. Okay, you call it ethanol amine. So that is the X group present in phosphatidyl ethanolamine. This is called cephalin. Then you have phosphatidyl serine. Serine is an amino acid. Okay, amino acid, you write it down like this, NH3. And then you have the alpha carbon, which is CH. Then you have the COOH. The R group here, the serine R group is what? It is CH2. CH2OH. Right? So where is this serine attached through this OH? Okay, this O will be in making a bond with this phosphate. Okay, so you can see here O, CH2, this one, CH, and then CO2 minus is actually a carboxylic acid group, and on the one hand you have amino group. Fine. So that is your phosphatidyl serine. Then there is phosphatidyl inositol. Inositol is a type of sugar. As you can see here, it is a hexose sugar. Then phosphatidyl glycerol. Phosphatidyl glycerol is nothing but it has another glycerol molecule attached like this. Okay, so that makes phosphatidyl glycerol. These are different types of phospholipids. We will study them again in biomolecules, but here I'm just showing you because it will be important later on in the next class or at the end of today's class. Okay. Now, after the model by Davison and Danieli, the sandwich model, which is also sometimes called trilamellar model. Lamella means layers, okay, and tri means three, so trilamellar model. There was another scientist called Robertson. He gave unit membrane model. This model was same as the previous one, except he said that there are glycoproteins present, not proteins. And he also said they are not globular. Okay, so if this is the phospholipid layer, then on top of them directly as a thick even layer you have proteins present. Okay, they are not globular, they are present as an even layer. Okay, so that is what difference he made to the original model. According to this model, he also said something else. He said the cellular and organellar membrane meaning the membranes of the organelles. They are structurally and functionally similar. Okay, structurally also and functionally also, the membranes of the cell and its organelles are same. However, similar. However, it is not always true. Okay, so both of these models, the sandwich model and the unit membrane model, are rejected because they fail to explain the selective permeability. Okay, cell membrane have this special ability called selective permeability. Selective matlab it can select for its own. Okay, it will select something specially, and permeability is to give permission to enter. Okay, so it can selectively allow only some things to enter. So this Robertson model or unit membrane model and the sandwich model could not explain how this is happening. Okay, then came the modern and the most widely accepted model called fluid mosaic model by Singer and Nicholson. Okay, as you can see here, this is a diagrammatical representation, but do you find it like a mosaic? Okay, mosaic is nothing but you must have seen somewhere that tile pieces are broken into smaller pieces, and these small pieces together they make some pattern or they make some drawing or painting. Okay, so if you do not know, Google this word mosaic, you will find out what it means. Basically it just means a patchwork, okay, of different types or different colors or different variety of things. Okay, so fluid mosaic model was proposed by Singer and Nicholson. According to this model, the proteins are arranged in the phospholipid layer as mosaic pattern. Okay, this is a phospholipid layer that you see, and in this phospholipid layer, like mosaic, you find different proteins to be present. Okay, these are all proteins, so they are present as mosaic. The model is easily explained as, if you want to explain this model, if you want to describe this model in simple words, how will you say? So it is protein iceberg. Iceberg, I hope you know, okay? The same thing which sank Titanic. Okay, the thing responsible for sinking of Titanic is called iceberg. Okay, so proteins are like icebergs. Some of them are just floating on the surface, while the others are embedded deep within. You know, tip of the iceberg, they say almost 2/3 of part of any iceberg is beneath, okay, underwater. So same thing is seen in case of proteins as well. Proteins are icebergs in the sea of phospholipid. The phospholipid is the sea and the proteins are icebergs, some of them floating, some of them deep within. Or another simpler example would be gulab jamun in a solution of sugar. Okay, in a sugar solution, you will see gulab jamun. Some of the gulab jamuns will be floating around, and some gulab jamun might have settled down. Okay, and then some of the gulab jamuns will be in the gravy, or not the gravy, but in the sugar solution. Okay, chashni. So in that chashni or in that sugar solution, the solution you can consider is made of phospholipid, and the gulab jamuns are the proteins. Right. So the fluid mosaic model, we will discuss this in detail. The phospholipids are rich in unsaturated fatty acid. Okay, we saw the structure of phospholipids. They have two fatty acid chains. So these fatty acid chains are unsaturated. Unsaturated matlab they have double bonds. Okay, because they have double bonds, they have a kink. Kink mane there is a bend. Okay, this also we will discuss in biomolecules in detail, but for now just understand that because of that kink their structure is a little bent. Okay, now because it is bent structure, they will not be able to be arranged in a uniform manner. Okay, they will not be arranged in a uniform manner, and this means that the membrane will be fluid. Okay, it will be liquid in nature. Also, you may know that unsaturated fatty acids are liquid at normal room temperature, right? So same thing apply here. Unsaturated fatty acids make the membrane fluid. Okay, right. If they were saturated fatty acid do not have a bend. Then in case of saturated fatty acid, they will be very uniformly arranged. And if they are uniformly arranged, then they will not be as liquid in nature or as fluid and as what you can say, um, yeah, as fluid in nature. Okay, now they are not actually fluid, so they are called quasi fluid. Let us understand that. Okay, quasi means false. Quasi fluid means false fluid. They are almost like fluid but not quite. Okay, so this quasi fluid nature of lipid enables protein to move within the bilayer. Now because there is a lot of space in between and they are fluid in nature, okay, quasi fluid in nature, the proteins which will be present in the membrane, they can easily move around. Okay, proteins will be able to move around. Not only proteins but even the phospholipids move around. Okay, for example, here is a phospholipid and here is another phospholipid. So if you see the diagram clearly, it is showing that this phospholipid can go and take this phospholipid's place and vice versa. Okay, so they can just swap places with the neighbors. Okay, change places with the neighbors. So this is lateral movement. In the same layer they can move around, or flip flop, which happens very less. Okay, so flip flop is from one layer the whole phospholipid will move down to the other layer. Okay, so phospholipids are also moving around freely, and in this proteins will also move around. If there is a protein here, it will be easily like floating. Okay, it will move around and it can come here or anywhere else. Fine. So this ability that proteins or phospholipids can move around easily, it is called fluidity. Okay, example or an experiment to show that fluidity exists. Think of this experiment. Okay, there is a mouse cell, and mouse cell has different phospholipids and it has proteins. These proteins are tagged by red colored dye. Okay, so that proteins become red colored. Then you take a human cell, and the human cell proteins are labeled with some green colored dye. Fine. Now artificially you are fusing the two cells. You are forcing the cells to fuse. As soon as they fuse, you will see a hybrid cell. The top portion will be of mouse, the bottom portion will be of humans. The time is 0 minutes. Okay, the fusion has just happened. If you keep it at 37 degrees and allow it to move around, okay, allow it to stay, allow it to rest, after 40 minutes you will see that the red colored proteins and the other green colored proteins have distributed evenly. Okay, they are almost randomly arranged everywhere instead of being separated. This means the protein can move around anywhere they want. Okay, and this shows fluidity of the cell membrane. This nature of membrane is important for various functions. Okay, this fluidity or fluid nature is important for growth. It is important for formation of intercellular junctions, for endocytosis, for secretion, for cell division, and a lot more things. We will discuss some of these terms in detail, so do not worry. Okay, chalo. Now, in this cell membrane, apart from phospholipid, there is also presence of cholesterol. Okay, cholesterol is also present, and wherever cholesterol is present, it decreases the fluidity. Okay, think of this spaces. Okay, because there is kink, there is bend, there will be space between here. So cholesterol will do what? It will take up this space. It will get arranged in the space like this. Okay, and because it is getting arranged in the space, then it will be almost like this, uniform. Okay, there will be no more space for it to be fluid. So as you increase cholesterol content, you decrease the fluidity of the cell membrane. Okay, but On the other hand, you increase the stability of the membrane. Okay, so cholesterol is important for cell membrane structure. It is absent in membrane of prokaryotes. Prokaryotes do not have cholesterol. Fine. Now we were talking about proteins, icebergs or gulab jamuns. Let us study them in detail. So there are two types of proteins present in plasma membrane. They can be integral or intrinsic. Intrinsic or integral, meaning they are a part of the cell membrane. Okay, they will be either partially like this or fully into the cell membrane. Okay, embedded into the cell membrane. They are tightly bound to the phospholipid. They are very tightly bound, so you cannot remove them easily from the cell membrane. When you separate the cell membrane for chemical analysis and study, you have to use detergent, because detergent can separate out the phospholipids, and then you are left with proteins and etc. There is a whole process. But if you have these intrinsic proteins, you will require stronger detergents to remove them from the cell membrane. Okay, this shows that they are integral, they are intrinsic, they are embedded into the membrane. They can be of two types: partially embedded, like this one here. Okay, it is only partly present inside the cell membrane, and partly it is outside. Or they can be totally buried or totally embedded, like you see here. Okay, totally embedded ones can be channel proteins, like a channel that is created here, which will allow things to pass through. Okay, or they could be other proteins as shown here, this egg shaped protein, right? We can have even some other functions, channel proteins and porins, like aquaporins allows transport of water, right? So this cell membrane, it is tightly arranged, but it will allow small molecules like gases, small non-polar molecules to pass through. Okay, non-polar, non, write it down, non-polar molecules will be easily able to pass through the cell membrane. Okay, as long as they are small in size. But for polar molecules which have charge or which are hydrophilic, you require some proteins, okay, for their transport. So they could be channel proteins, they could be carrier proteins, they could be porins, etc. So these are the functions of the proteins. They can be helpful in transport. As you see here, something is getting transported from outside to inside or inside to outside. Then they can do enzymatic activity. Some enzymes are fixed in the membrane. Okay, I'm not talking about cell membrane, but I'll give you an example of succinate dehydrogenase. Succinate dehydrogenase is an enzyme of TCA cycle, which is present in the inner mitochondrial membrane. Okay, it is membrane bound enzyme. So same way, even in your cell membrane, you find some enzymes which are embedded. Okay. Then for signal transduction meaning there will be some signal coming from outside. It will attach here, and that signal will be passed on to the cell inside. Okay, will be passed on to the DNA, to the RNA, and to the specific organelles. Then it is important for cell recognition. Okay, these proteins will help in recognizing other cells, intercellular joining, and attachment of cytoskeletal and extracellular matrix. We will discuss these few of these parts later in the lecture. Okay, do not worry. Right, so that was all about integral or intrinsic proteins. Then the second type of protein, the floating ones. Okay, the gulab jamuns that were floating, they are peripheral or extrinsic protein. Fine. So peripheral proteins, again let us consider this RBC membrane. So these proteins, what you see, okay, this protein is called spectrin. So spectrin is a peripheral protein. It is present on top of the cell membrane. Okay, Here it is shown farther away to make the diagram clearer, but actually it is present very close to the membrane like that. Okay, it provides structural support and shape to the cell. Fine. So that is the role of spectrin. Spectrin is a peripheral meaning in the periphery, or extrinsic meaning outside protein. They are superficially arranged. They are not inside the phospholipid membrane, but they are just present on top of the membrane and can be separated easily. The proteins are hydrophilic because they are present either outside or towards inside, towards cytosol, which is also hydrophilic, and outside is also hydrophilic. Okay, so these proteins are comparatively hydrophilic and they can be removed easily, so they are not permanent. Some of them may be permanent, but not all of them are permanent. Okay, these proteins have enzymatic or receptor activity. Example is spectrin protein that you see in this RBC. Apart from proteins and phospholipids, the membrane also has carbohydrates, only 1% of total cell membrane, but still. So these carbohydrates are in form of, they're not present just like that, okay? They are present in form of glycoproteins or glycolipids. Here is what a glycolipid looks like, okay? It is a lipid which has this blue color means sugar, okay? So it has sugar attached at the top. Remember that phospholipid which has phosphate. Now that phosphate group can get attached to a few units of sugars, okay, and then it will form a glycolipid. Same way, this is a protein, and on the protein there are some such branches of carbohydrates. Okay, you can see a lot of carbohydrates present on the protein. Such proteins are called glycoproteins. Okay, so glycoproteins and glycolipids, they have carbohydrate moiety on them, as you see here. This here is a peripheral protein, other all are integral protein or intrinsic proteins. Fine. Now, why are these glycolipids or glycoproteins important? So they are important for cell recognition. For one cell to recognize other, you require glycoproteins and glycolipids. Okay. Example, during fertilization, the sperm and the egg recognize each other because of these glycoproteins. Okay, these glycoproteins, for example, if this is egg and this is the egg's glycoprotein, the sperm will have an opposite receptor. Okay, it will be able to receive something. For example, this will come closer and then it will get fixed here. Okay, and then it is able to sense this glycoprotein and come to know that okay, this here is an egg or egg cell. Fine. So sperm and egg recognize each other because of these glycoproteins. Then blood group antigens. You have blood group A, you have blood group B, you have blood group AB. O is absence of antigen. Okay, O is actually 0, meaning 0 antigens. So blood group antigens AB and AB are because of the glycoproteins. Okay. And the interaction between such glycoproteins and antibodies happens because of the glycoprotein, okay, on the RBC. Then receptor mediated endocytosis happens because of such glycoproteins. This again we will be seeing later on, but just for now, understand it like this. This is your cell membrane. It has some receptors. Okay, receptors are protein molecules which receive some signal or which sense something. Okay, now let us say some bacteria is coming, and on the bacteria they have some glycolipid molecule. As soon as it will come here, the bacteria, it will get fixed here. Okay, there will be recognition of this glycoprotein. Now what will happen? It will start doing this endocytosis. So now you have this cell receptor, you have the glycoprotein, and you have the bacteria. This bacteria will be taken inside the cell. Okay, it will be taken like this. Okay, here again receptor and the bacteria. So this is how receptor mediated. So there is, because of receptor, there was endocytosis. Endo means inside, cytosis means to take something inside the cell. Here we end today's class. We will continue with the cell membrane in the next class. Okay, some detailed functions and whatever we discussed, endocytosis, exocytosis, transport, we will discuss in the next class. Okay, see you next time.